A very good evening to all of you. Uh, Welcome to the final session of the topic of nucleic acids. This is the fourth topic of this series where we learn about the function of nucleic acids. Uh, in this particular session, uh, we learn here we learn and explore how DNA and RNA work together to create proteins, the machinery of life, and the learning objectives uh, of this session will be DNA replication, transcription, translation, regulation of gene expression are the basic objectives of the session. When you look ahead, when you say with the functions of nucleic acids, if you look at the functions of nucleic acids, they play very, very vital role in functioning, reproduction, survival of living organisms. The primary functions include genetic information storage. DNA stores the genetic information, and it's the blueprint for all the living organisms. It contains instructions for the development, growth, reproduction, and repair of cells. The genetic information transmission of DNA replicates itself to pass genetic information from one generation to the next generation during reproduction, which ensures the continuity of life and heredity. One of the most important functions of nucleic acids is protein synthesis. Um, the RNA translates the genetic information from DNA into proteins, the functional molecules in cells. And in the sequence, what we look at it is messenger RNA. What is the function of messenger RNA? It carries a genetic instruction from DNA to ribosomes. Transfer RNA delivers amino acids to ribosomes for protein assembly. Similarly, the ribosomal RNA forms the structural and functional components of ribosomes. Regulation of gene expression, RNA molecules regulate genes which are turned on or off, influence cell behavior and function. Examples examples include microRNA, small RNA interfering RNA, energy transfer where ATP is transferred. Uh, Nucleotides, the building blocks of nucleic acids, serve as energy carriers. Adenosine triphosphate, a nucleotide that stores and transfers energy for cellular processes. Uh, One of the smaller function of nucleic acids are enzymatic and catalytic functions. Some RNA molecules uh, called ribozymes act as enzymes to catalyze biochemical reactions such as peptide bonds, formation of during protein synthesis, cellular communication. It's one of the most important one. Cyclic nucleotides like cyclic AMP, we generally call it as CAM, are signaling molecules involved in cellular communication and regulation of metabolic pathways. Now, these are some of the primary functions of nucleic acids. And when we look at central dogma, if you look at central dogma, the central dogma DNA makes RNA and RNA makes proteins. This flow information drives biology. And if you look at replication is a semi-conservative process. Each new DNA strand contains one parent and one new strand. What is the role of a DNA polymerase? If I look at it, DNA polymerase ensures accuracy. Now, in the transcription part, RNA polymerase synthesize messenger RNA using DNA as a template. Messenger RNA carries instructions for protein synthesis. This we call it as a transcription. There are two parts in transcription, generalizing as the slide goes by: DNA to RNA, and messenger RNA carries instructions for protein synthesis. So the second part is only protein synthesis. If you look at translation occur in ribosomes. Messenger RNA codons are read, and transfer RNA brings amino acids to form polypeptides. This is one example of a genetic code. Genetic code means triplet codons correspond to amino acids. It is universal, redundant, and precise. If you look to explain about it, if you look to explain about it, if I take the peripheral, the white ring is the amino acids. Now the inner first ring are the bases which are getting paired with amino acids. The inner ring is the one which is having a triplet codon. If you look at very finely, if I take the first quadrant, I have uracil, cytosine, adenine, guanine. Four of them are coded with U, and those can be corresponding with respect to leucine and phenylalanine of the amino acids here. Codon means three, so three of them can bind together with one amino acid here. Now this is one amino acid codon wheel, which can be represented. It's like a wheel where we start from this particular end, spin it around, and it stops at one particular point, and that particular point binds with the corresponding amino acids and making it a codon. Now, moving ahead, we shall use the word regulation. Genetic expression is regulated by promoters, enhancers, repressors, and epigenetic modifications. Here, we try to answer a reflective question from what we learned and what we have seen in this particular module. How can a single DNA mutation change a protein function? Think of diseases like sickle cell anemia as an example and address this particular reflective question here. And there is also to be answered is a reflective discussion. At the end of the session, a link will be provided for the discussion, which is open for all the students. Please do participate in the discussion and encourage all others to write uh, freely for the discussion part of it. And each student has to at least answer for two more students queries or answers or responses, what they think about the reflective question here. Now, moving ahead is a reflective uh, discussion. In the reflective discussion, discuss how DNA mutations lead to protein structure and functional changes, which is both harmful and beneficial. How do we justify this statement? Initiate a discussion and do complete about it. In the concluding part of the particular module, of this particular module, we always see nucleic acids serve as the command center or the directing growth and inheritance. That's the final conclusion what we have of this particular module. And for your reference, all the uh, module synthesis, module extracts are taken from either Biology, OpenStax Biology, NPTEL, EG Padashala, and PubChem. are the various sources which have been sourced for the content part of it. Thank you for being the part of the interactive session. Hope you enjoyed the session very much, and feel free to ask additions, post uh, questions and comments on the link which has been given below. See you next week with one more module of interactive session. Assignment has been created. Please do answer the assignment and also participate in the quiz. Thank you.